saying record. See you guys over here. And get out the chat and get out the participants. Zoom has a lot of pros, and they really do have a lot of pros. But one of their major cons is when you share your screen, it puts everything on top of itself, so you can't find anything. So the reason it takes me a few seconds of delay is it puts the screen where I can see your face directly on top of the participant list, which I need to see in case someone gets kicked out and they're trying to get back in, and directly on top of the chat, which we also need to see in case someone types in an answer. So it's a really, really clever design. So that's a long way of saying that. All right, two-way tests. So now I apologize. We have two sample tests. We have two-way tests. We have one sample test. We have one-way test, also known as K-sample test. These words get confusing. Don't try to memorize the words because it's not really that interesting. What we're talking about here is we're talking about two-way ANOVAs. That is my dog, so I'm going to switch over to headphones.
All okay.
So can you guys hear me? Okay, good. A two-way test, very simply, is an extension of a one-way test, where in a one-way test, you're looking at a single factor. And we looked at state relative to median income. We looked at education level relative to lifespan. With a two-way test, what you're looking at is you're looking at two separate variables. So you may look at both education and gender on lifetime earnings. I don't know if you still are getting off on the election, but probably the most common demographic I keep hearing about is a relationship between education and gender, or gender and location in terms of how individuals voted in the presidential election. In those cases, these are two separate variables that we're looking at collectively in order to understand their overall influence on a third variable. The effect of smoking and political perspective on weekly exercise. I actually thought that was an hysterical study, but it's true, where they looked at sort of whether a person smoked or not relative to whether they were conservative or progressive, and then how that influenced the amount of exercise they did per week. Who would have thunk it? The effect of detergents and water temperature on the amount of soil being removed. Each one of these, we have two variables, two categorical variables, and their influence on a third variable that's continuous. An example that maybe is more of biological interest, these are white-crowned sparrows. White-crowned sparrows are famous because they were the first non-human species that demonstrated culture. Each population of these birds sings a location-specific song that they learned during a developmental period. So like culture, these animals learn something that is specific to their location. And one of the things that they looked at is they looked at the relationship between sex, male versus female, relative to food stress versus unlimited food in the development of different areas within the brain that are associated with song learning and song production. In some cases, there is no overall effect. In other cases, there is a strong sex effect. And then in some cases, there is a sex effect as well as a developmental effect in terms of the animal's ability to produce these songs. Mathematically, this is what we have. And you can kind of see that this looks like an ANOVA, a K-sample test, where we can predict an individual measurement based on the overall mean of the population relative to what are called main effects. This is the first variable. This is the second variable, and potentially an interaction term. So the first variable might explain the overall phenomena. The second variable might explain the overall phenomena. And then there might be an interaction between those two variables that explain the overall phenomena, so that your observations are the mean of the measurement samples relative to the error. And does error mean in this context? Not all at once. No, I'm getting my Google Drive. So I'm going to go with Ben. Ben, I don't see your camera on, which is why I picked anybody.
All right. I am not sure.
Totally
Yeah.
fair. What does error mean? It seems like a bad word, doesn't it? The error. Because what does error normally mean?
Chance to be wrong.
The chance to be wrong. Makes perfect sense. It's a great way of describing it, Ben. Normally, error reflects something about being wrong. That's not the case when we're talking about statistics. Error is not a term associated with a mistake. Heather, you're actually first up on my list.
Of course.
So if it's not
Does it have to
chance
do
of
with
being a mistake, what might it be?
like outliers?
Yeah, not so much outliers. Not so much outliers, but I like that idea. So it's not outliers. It's
Is
not
it like
making
the
a mistake.
difference?
It's the
between
other
things?
variables. So I think Maisie, you're getting at it. It's the other variables that are not being measured that are influencing your observation. So if we go back here, are education and gender the only two things that are going to influence lifetime earning? The answer is obviously not. We already know residency, what state you're from, influences lifetime learning. And there's probably tens, maybe 100 additional variables that all go into influencing lifetime learning. In the equation where you're looking at gender and education, all those other variables, that's your error. In the case of weekly exercise, are the only two things that are going to influence weekly exercise smoking and your political perspective? The answer is no. There's a ton of other things that are going to influence weekly exercise. That's your error. That's your error. What is called the process error. The other things that are influencing the variable of interest. Make sense? So when we're looking at the overall population mean, which is relative to the main effects, and we have two of them because it's a two-way test, and potentially their interaction. You sometimes look at this, you sometimes do not. For two-way tests, more often than not, we tend to. So what do we mean by that? Well, here's some different effects. If we have this basic equation, and this is our first main effect, that's what this might look like visually, where one set of the x-axis are distinctly different than the other set of the x-axis. In one case, the bars on the left are larger than the bars on the right. In the other case, the bars on the left are smaller than the bars on the right. These two groups are distinctly different. The other main effect is comparing within those groups, where maybe in this case, the green bars are significantly larger than the blue bars, or the blue bars are significantly larger than the green bars. And then the last are interaction terms. The interaction term is not so much necessarily that your A variable is different or that your B variable is different, but that in the first condition, B is larger than green, but in the second condition, blue is larger than green, or blue is smaller than green. And then of course, there's the alternative option where in the first condition, blue is smaller than green, and in the second condition, blue is larger than green. So if we go back here, we can see how this is no effect. Basically, all of our bars are about the same. They have the same medians. They have the same error terms. Here, we have a main effect, and we have a first level main effect. So males have a larger, what is called RA, than females. Here, we have the same type of main effect, where males have a larger area X. And that is literally what the location of brain is called, area X. It kind of reminds me of Planet X from the old Looney Tune commercials, but that is it. Females have a relatively small area X. But here, HVC, high vocalization center, this part of the brain, we have an interaction term, so that there is a dramatic decline in the area of HVC in males when they're food stressed. But in females, when they're food stressed, there's not a dramatic decline. In fact, statistically, they're really about the same. So that when males are food stressed, you see a lower volume, but when females are food stressed, they're still about the same size as a female. So an interaction term. Do those three properties make sense? Our first order main effect, second order main effect, and then what we could see for possible interaction terms. All right, so we're going to play with these data, and I still like these data. They're a little bit long in the tooth in the sense I think this was 2013, but in 2013, the food forest brought in a massive pile of organic compost for the garden. And how many people have been out to food forest? Know what I'm talking about? Yeah, it's a great spot. If you haven't been out there, I would encourage you. If nothing else, it's a nice garden. So it's just a nice place to kind of just be around some plants and just sort of enjoy the outdoors. 10 years ago or so, it was a little refuse dump. It was just random debris. There was no organization. There was nothing really involved. Students got together, they create what is a food forest, and that is a mixed use agricultural plot with different plants of different variety. And they grew it to be an organic, what is called a permaculture. It's part of a lot of students' service learning. A lot of people in colloquium will go out there, and then a lot of students just volunteer there in general. So not surprisingly, if they're gonna get mulch, they wanted organic mulch. Who came up with this sort of design? I don't know, but they put the food forest directly next to the Kleist Center for Health Education. And how many people have been to the Kleist Center? Okay, a few. And did you go as university students or did you go as K through 12?
University.
Okay. Well, the Kleist Center is one of our biggest outreach programs for local elementary, middle school, and high school students, where they come onto campus to learn about different health issues. So you have the food forest here. Right next to it, you have the Kleist Center. In fact, I think I can illustrate that. Yeah, here. You know, it's a crude illustration, but it kind of shows the point. So food forest brings in this mulch. This mulch contains large population of two invasive millipedes. One is the rusty millipede from Thailand. The other one is a black and yellow banded millipede from the Bahamas. And these millipedes did very well. And these millipedes moved across the food forest, potentially, and they moved into the Kleist Center, so that for about 2 or 3 months, the Kleist Center was completely covered with millipedes. And they didn't like that, like not at all. And the millipedes got inside the building, so when you would go in there on a Monday, the entire floor was covered with millipedes, which they did not like. They did not like at all. So what did the Kleist Center want to do? Any thoughts, any ideas?
Kill them.
They wanted to kill the millipede. And for people who do not maybe value things like the food forest, how do most people kill arthropods?
Birds.
Oh, you could do birds. You could do birds. I like that.
Poison.
Poison. Most people just want to dump down a lot of poison. And is that consistent with the goals of the food forest? No. So there was a little conflict between the food forest and the Kleist Center about one, who's responsible for these millipedes, and two, how are we going to control them? And three, is that control going to be consistent with the idea of kid health and permaculture in the food forest? Kind of make sense? So we did a study, and we did a two-way study. And what we were interested in doing is we were interested in looking at two things. One, we were interested in looking at where millipedes were being found. And how we did this is we created what are called pitfall traps. They're very simple little sampling technique where you put a pit in the ground and animals walk into it. And it gives you an estimate about how many animals are in a location and where those animals are. So if you get most of your animals from location number 5, then that suggests the millipedes are coming from the Kleist Center. If you get most of your animals from like location 1 or 2, that suggests they're coming from the food forest. We also wanted to look at the effect of light, because one of the things we noticed at the Kleist Center is that they seem to congregate right on a light that was directly in front of the door closest to the food forest. So for that, we set up each one of these pitfall traps in a pair design where one of them had a light that was turned on and the other one had a light that was kept off. So one pitfall was trapped, one pitfall was lighted. Make sense? Two-way design, location and light. So what I want you guys to do is I want you guys to load the data set. And I want you to take a look at two different models. One is going to be a model that looks at just the main effects. Oops. If you put a plus in between your two main effects, then the model is just going to look at the effect of one variable, z, and the second variable, z. If you put a multiplication symbol between those two main effects, then it's going to look at the effect of z, the effect of x, the effect of z, as well as the interaction between those two variables. In addition, I want you to tell us the difference between the two procedures. Use the model that says, actually, I have to check that real quickly. I think I might be giving you the wrong instruction there. So give me 1/2 a second. Okay, use a model that says millipedes. Do not use proportion. Any questions? Okay, I'm gonna send you guys out into breakout rooms to get working on this.
So you said to use a question that said what?
Use the variable millipede. Do not use the variable proportion.
Okay, thank you.
Empty Alana, yeah, guys, let's head on out.
Sometimes it feels like we'll get confused or something. As you guys go through the groups, you'll clarify something. So we use those last 30 seconds as wait, we had an idea.
No, it's good. Do. That's what you should do. All right. So, Ryan, I'm not sure I saw Ryan yet. Stephanie, I saw Stephanie, right? Yeah. So, Stephanie, up here, I created some code to get us started. I've run nothing yet. So, by the way, at this point, I would load those two libraries to begin with always. Some of the other packages, we'll need them. Maybe we may not. It depends. But these two are pretty much universal. HMIS allows us to use Describe, and ggplot2 is how we're going to be plotting for the rest of the semester, except for circular data. That'll be the one exception. So every time, toss those up. I just copy and pasted these over, so I have a new script. And now I'm going over here, two-way, grabbing millipede. I deleted the proportion from my data sheet and I re-uploaded it onto Canvas. So if you want to, you can grab the same one. If you have proportion, it's no big deal. We do the attach.
Quick question.
Yeah.
Do we actually have to load ggplot, because under HMISC it says loading required package ggplot2. So are you already importing it?
Never noticed that. It's a good question. Okay. Try it, see what happens.
All right.
Yeah, good observation. So run attach. Tristan, why don't we do attach?
So that R knows the different headers and stuff.
Yeah, perfect, perfect. You do not have to do it, but it is tedious to write out the matrix, $, variable name each time from my perspective. So I always use the attach. So now, Stephanie, I'm coming to you. Why did I do summary and what do I see down here that I need to attend to?
I don't know. I did summary too, but I didn't look at anything.
This is a new thing this semester because of the update in R. Does anyone remember why we're doing summary and what we're looking at in the result? Heather?
It says like class and mode are characters.
Yeah.
So you have to
Yeah,
change.
so go ahead, sorry.
No, it's okay. You have to change them to factors.
We need to change them to factors. So it's not reading, treatment, and
Okay.
location, and this is inconsistent. If you're able to run these without changing the factors, do it. But for a lot of us, based on whatever version we have of R at this point, we have to go through this additional step. So we go up here, we do location equals factor of location, and we do treatment equals factor of treatment. And then I'm obviously going to want to run one more line of code. What's that?
Summary again.
I like to do the summary again to make sure that everything is working. So we do those 2 steps and it's still not showing us, but we'll try anyway, see what happens. All right, we do describe. We can see that we have different locations, both location in between the food farce and Kleist, the food farce, Kleist, and then we have 2 different conditions for treatment, light and dark, and then millipede. What type of data is millipede? Cortland.
Continuous.
It's continuous. So our Y variable is continuous. All right. So now, Morgan, you're up.
Yeah.
I want to look at a one-way test. Tell me what to type.
One-way test? Is that whenever?
No, I'm sorry, I lied, I'm stupid. I want to look at just the main effects, just the main effects. I don't want to look at the interaction.
Oh, okay, so you have to do the variable underscore me equals AOV.
Okay, yep, so let me write the fit me. I'm gonna change it to fit me if that's okay.
Yeah, that's fine.
AOV, and then talk to me. What am I doing now?
And so I put millipedes.
Okay.
And then I did the squiggly
Yep.
line, sorry. And then I did location plus treatment, and then close the parentheses. And then under that, I wrote summary. And then in parentheses, I did variable underscore me, ME.
Perfect. Perfect. Okay, so one of the things to note, the order matters. Since you are partitioning out variation, the first variable is going to be the first order of variation. The order to which you put these will affect your answer. So what we generally do is we put the more important factor for our research question first. And then the less important factor for our research question second. In this situation, why are we doing location first and treatment second? To which I agree, Morgan, that's how I would have set it up. Any thoughts as to why?
Maybe because we have multiple treatments at one location, but I think that would work. Like you have both light and dark at each location.
We have light and dark at each location. It has nothing to do with science.
We're just trying to figure out we're trying to figure out where the millipedes are coming from.
Bingo. And each party wanted to accuse the other. So we're just trying to figure out where the millipedes are coming from as evidenced by where they're more common. And considering this was a conflict between those 2 places, We just want to find that location. That was the more important thing from their perspective, which is why we treat it first. So we run these, we look at our fit, and this is what we get. All right, so a little quick piece of advice. I would copy and paste these out. I would go over to Excel. I would open up a new sheet. And it's on the wrong window, so let's put it over here. And I would paste it. Sometimes it might paste properly into worksheets, sometimes it does not. So I go over to data, I do text to columns, I do next, and I look at how it's defining and it's breaking up my columns very nicely. I do finish. So then I can then have information. Now, where do we start looking? Rotilla, you are up.
Yes, sorry, I didn't understand the question very well.
Great. We have results on this spreadsheet now. We have results down here in R. They say the same thing. What I want to know is now that we have some results, where do we want to start looking to see if there are any significant patterns?
P-value?
P-value. Okay, so great. On this spreadsheet, where do I look?
Last column, F column, I think.
Not the F column. We're going to get to the F column, but we look at these two. So we have a p-value for location, and we have a p-value for treatment.
Yes, that's what I meant.
Sure. So if I were you guys, I would highlight them because you're going to need to use them. And I like to highlight those things that I'm going to need.
Hey,
So
Professor,
what can we see? Go ahead.
do you get them organized like that again in the Excel file?
Sure. So let's actually do it again. Not going to save that. What I do over here is I go over to R, I copy it out. I like to open up a new worksheet in Excel, which I lost again. Paste it. Right now, it's pasted everything in the same column. So if I highlight the column, I can go to data. I go to text to columns. And then R is going to try to separate these out into individual cells. What R does is show you where those new row columns are going to be with these lines and arrows on it. If they match up to what you like, great. If they don't, you can double click it to get rid of it and double click it to remake it.
Oh, so you
It'll
can like,
put, go ahead.
you can like move the arrows, right?
Mm-hmm. Mm-hmm. You can put them anywhere you want. So I want to make sure that my columns are going to make sense. Here's my variables, degrees of freedom, sum of mean squares, mean sum of squares, F values, P values, and these little dots at the end. Once it all looks good, I hit finish. And now everything is spread out nicely and organized well. We need to start with our p-values. I'll highlight them. So now I would like Forrest. You're up, buddy.
Oh boy.
Result sentence for location.
So P is, I guess, 0.01. So we have a significant difference between locations, I guess.
Okay, okay, but we're biologists. Back to this image.
So yeah, we would say that there is a difference between is this based on concentration, like
the number of millipedes?
Of millipedes.
So what you're counting is the number of millipedes you found in the pitfall trap at the food forest location versus the Kleist location versus both.
Okay. So since we don't know which way it goes,
we don't know which way it goes yet.
We would say there is a difference in concentration of millipedes between both sites.
And you have two choices. You can use in Excel if you like, or we can go back to R. And you may just want to put this in your script. And therefore, you may just want to copy your results into your script. My one thought about that is it doesn't always organize information as clearly. And therefore, you have to do a little bit of extra work to make sure everything aligns properly the way you would want to see it. So far like that. Now, Forrest, what information are we missing in your sentence? There's a difference in the concentration of millipedes between sites.
Yeah, so we need the, I'm trying to remember for ANOVA, we need the F value for ANOVA, right? P value
so
and degrees of freedom.
you have two choices. You can write table 1, and therefore people just go to a table that you will have created that shows all this information, or you can do F equals 4.64 DF, and there's 2 degrees of freedom, 2,112. P equals 0.012. And it's important to remember that there are 2 degrees of freedom here. 1 degree of freedom is associated with your variable, and the other degree of freedom is associated with your sample size, the error term. So now, Casey, you're up. I want our result for treatment. Casey went blank for a second. I will come back to her. Isaac.
I'm so sorry. I had just come back from the bathroom and I heard you say my name.
Not a problem, Casey. I'll come back to you in a second, okay?
All right,
Isaac,
thank you.
Yes.
give me my result sentence for treatment.
I do not know. So,
Well, let's start with the p-value. Is that a small or large p-value?
oh, it's a small p-value.
It's a very small p-value. So are the treatments the same or are the treatments different based on a small p-value?
They'd be the same.
What's the null hypothesis?
That there's a difference. Okay.
The null hypothesis is that there's no difference. And when there's a small p-value, what can we do to the null?
Reject.
We can reject it. So give me a sentence.
So there is a difference. Okay.
I'll start with that.
Based on this, there's a difference between treatment and the number of millipedes
Okay.
between both sites, or between.
Well, we're not going to worry about the site yet.
between treatments. Yep.
Treatments and the number of millipedes. Okay, and now give me my evidence.
Your F value is 15.41. Degrees of freedom is 1.
Degrees of freedom is 2
And-
numbers.
Oh. What am I looking at here?
The degrees of freedom is always in an ANOVA, including a two-way ANOVA. It's both the degrees of freedom for the effect and the degrees of freedom for the error. So you need
Gotcha.
to know both
Okay.
the effect as well as the sample.
Okay.
And then what's my last part?
Last part is your p-value, which is 0.00015.
And what do we round that to?
Just 0.001.
Yeah,
Zero,
less than
zero, zero,
0.001.
Yeah.
Yep. Okay, beautiful. Beautiful. So this looks good. Casey, you're up now. I want you to tell me the model we need to run to look at for an interaction term, because this only looks at the main effect. Notice we're looking at location and we're looking at treatment, and only location and treatment. We're not looking at the interaction between the two. So I want our next model.
So our next one would be the two-way test for interactions.
Okay.
So that would be fit underscore, however you want to do interactions. So INTX was used. So you could do
Yep.
INTX. And the only thing you're going to change with that is you're going to change the plus to a asterisk, the little star symbol, that. And then, of course, in the summary, you're going to do fit underscore INTX.
Great.
And then if
So
you run it,
we run it and we get this. So let's put up our results. There we go. Are the p-values for location and treatment exactly the same as they were for the main effects? No, they change. Any reason as to why they might change? Reason is
Interactions.
now we're, go ahead, sorry.
I was gonna say interactions between variables.
You get, because now we're also looking at the variation as a result of the interactions. So that changes all the p-values in the analysis. So when you make a choice, you generally have to choose one or the other. More often than not, in fact, there's very few situations in a two-way test where you're just going to look at main effects. Pretty much in all two-way tests, you also want to look at the interactions. And do I see Dakota? I don't think I see Dakota yet. Alana, very quickly take me through. Is there a difference based on location?
There is a difference based on location.
Perfect. Is there a difference based on treatment?
There is.
There is. Is there a difference based on the interaction between location and treatment?
Oh, there is.
There is? Is that a large p-value or a small p-value?
My glasses ain't that good. Hold
Oh,
on.
sorry. Let me give me a bigger,
Okay.
bigger. Is that better?
Mm-hmm.
Okay.
It's a large p-value.
It's a large p-value. So is there a difference or not?
There's no difference.
There is no difference. So there is no interaction term. So there is a difference in terms of location. There is a difference in terms of treatment. There is no difference in terms of interaction term. Make sense? All right. So this is what I want you guys to do, because let me go back here for 1/2 a second. I don't remember who said it at this point in time. I actually think it was Forrest, but is this a very satisfying sentence to say there's a difference in the concentration of millipedes? Or do you want to know where the millipedes came from? I want to know where they came from. And then, is this very satisfying? There is an effective treatment. Or do you want to know which actually catches millipedes, light or dark? Light or dark. So let's go find out. So for that, what is our best route? And Riley, what is the best way we could go about finding out what the actual pattern is? We know there's a difference in some place. We know there's no difference in other place. So how can we go find a pattern? What should we do?
Make a graph or plot.
Ah, perfect. That's exactly right. So you guys are out of here. I want you guys to use ggplot2 to make appropriate bar plots or box plots, your choice. Make sense? Questions? Okay, let's open up your rooms. Let's recreate them. Actually, let's recreate. See you in a bit. And Riley, I think you're up. So where's the mistake in my code at this point? Like really kind of dangerous mistake.
I don't see any code.
Oh, because I'm not showing anything. I'm not showing anything?
No, I see your screen. It's just I see like lines 34 through 41. And it's
Oh,
all
you
like,
can still see the problem.
I don't see it. Is it just you're assuming you're making a bar plot?
No. You're only going to make this mistake a few times. And this mistake, like when I have to suffer the consequences of this mistake, I lose my mind. It is irrational.
You haven't saved.
I haven't saved anything. I hate losing my progress. It drives me crazy.
I was gonna say that next, by the way.
Got you, Ryan. Riley. Sorry.
Good save.
Two-way.
Two, Heather.
Ryan.
Yes. All right. So, Ryan, so we're coming back to you, buddy. So I want us to create a bar plot. Get me started.
Me?
Ryan, you're up after this. Yeah.
Oh, jeez.
Me?
Yes, sir.
Sorry. Okay. So let me see. I wrote milliplot equals ggplot, parentheses, AES, X equals location, Y equals millipedes. I'll put a comma after location. And millipedes. Fill equals, what was it, treatment?
All right, what's going on at this step? What's going on with fill?
I don't really know how to explain it, but basically it shows both kind of treatments on the same plot. Like it shows the difference between the two treatments on the same plot.
Okay. It allows you to show both treatments. Can anyone see where there's a mistake? And we know there's a mistake because we got this big old, uh-oh.
Open parentheses before the X is equal to.
Yep. Yeah, open parentheses here, and therefore we need a close parentheses there. Okay.
And then
anything else?
Yeah, comma in
Okay.
between those. Data equals whatever you name.
I don't know what I called it. I called it mill. So we all good with that? So the main difference between the one-way plot and the two-way plot is the fill. We're going to change the parameter of fill so that it's the second variable. Our x-axis is going to be one variable, and then how we color the bars is going to be the second variable. And we'll see what that looks like in just a few seconds. So can someone tell me what to do with the bar plot now? Oh yeah, that's Ryan. Hey, Ryan.
Hey.
So Roddy got us caught up to sort of set up the parameters. And if you remember, if we run this right now, we do, nothing happens. So if I do milliplot,
You millplot. And you go stat underscore summary. But you want to do the
we get this, but that's not telling us anything yet. So how do we actually create a plot?
You use stat underscore summary.
Okay, so I go milliplot stat underscore summary.
Yeah, and then you're gonna use GOM or open parentheses GOM equals and then you're gonna use these colon semicolon or and you're gonna put bar and close it and then put a comma after it
Okay,
and then put fun equals mean.
with quotes?
Might
Oh,
I
no.
intervene and just suggest we copy and paste from last class or 2 classes ago?
Oh, I'm
Just
okay
so
with
he
that.
doesn't.
Or you could do that, I was just being extra.
You mean like this?
Yeah.
Okay, okay, I like that. I like that. So let's go back over here. That's pretty clever. The only thing, Ryan, that you had set up so far that was a mistake is we need a plus.
Oh, okay.
Someone remind me what the plus does in every case?
Is it adds to?
Yeah,
What you're
it's
just doing?
basically adding another piece of graphical grammar. So we have the parameters of our plot. The next thing we're going to do is we're going to add in the bars. And then we have another plus. And then we're going to add in the error bars. And then we have another plus. And we're going to add this theme thing. Should we run it? Everything look good? We got
Yeah.
dodge, we got error bars, we got dodge, we got width, we got theme. What do you guys think? Should we go for it?
Why not?
Why
What's the
not?
worst that can happen?
I don't know. We'll find out together. Is that pretty okay?
Beautiful.
Yeah,
Is it beautiful?
Not really.
the error bars.
It's not
error
really
bars.
beautiful.
Looks kinda
yet. It's not ugly, but it's not beautiful. Okay, so let's start with the first order problem. And Tristan, buddy, you're up for this.
Hello.
What is this coral and what is this blue? Do we know what these refer to?
Those would be different groups. I imagine
would
that's
be
the
different
light
groups.
versus not light.
That's exactly right. That's our treatment. That's our light versus, and I like the way you put that. That is light versus not light. I enjoy that. Or light versus dark, but one of those two, they both get us to the same endpoint. That's awesome. Why aren't we seeing the names for these? What in our code has deleted that reference?
Dodge or legend position none.
Ah, bingo, bingo, bingo. So let's get rid of that. Because we're not going to use that this time. We actually want to see it. So what about this plus sign, Tristan? Last question for you. Do we need to
You
keep
get rid
it?
of that. No,
We get rid of it. If we leave it,
Yeah.
we're going to get an error. Why?
because we're not adding anything after it.
We're not adding it. Or it's going to expect, what do you want to add? And you're going to say nothing. So it's going to get confused. So we delete that. So let's run that. Okay, that looks better, right? Now we know what these two bars refer to. Blue is for light, dark is coral. We noticed, what did we notice about the light? So do millipedes go to light or do they go to dark?
To light.
Oh yeah, they did. Oh yeah, they did, dramatically. And where do we find most of the millipedes? We find them in between the clay center and the fruit forest. We find a lot of millipedes in the fruit forest. We find less at the clay center. So it's really kind of in between. So are these figures good yet?
No.
Anyone have some, we have to fix these error bars. Anyone have some code where they came up with some ideas? Forrest, I think you were working on this. What did you come up with?
The closest I got, and still messing with it, is if you remove width, it will center them, but then it makes them the width of the bars so that it looks like dumb.
Okay, let's find out.
And I hate it, and I don't know how to fix it.
I think that is totally fair. That is totally fine. But it does allow you to at least give me something that is not inaccurate for an homework. And you could therefore claim, well, maybe Gunnels is not totally aesthetic, but it's not inaccurate. How do you go figure out the solution to that?
Help.
You got to Google. ggplot2 error bars too thick. And so we come up with unable to adjust the width of the error bar. I like that. And we get some code there, and I don't like that. And I go width. I kind of know that's not going to work, but we could try it. And that reduces, but it doesn't center. So we go back to Google and we try ggplot2. Error bars not centered. So we get here. Problem placing the bars in the center of columns. We get a lot of stuff there, but that's the person's problem. Oh, they're showing us their problem. So that kind of looks like our problem. Now we can see, do we get anything here? Identity dodge. That identity, if you have a problem with the G error. Position dodge, that's not helping us. Oh, that person's persistent on this. Ooh, maybe this. We're going to ours. Let's see what happens. Yeah, it didn't like that. Oh, because we already had a position of dodge. That doesn't look any good. So I don't have an answer yet, but that's how you go about finding out stuff. You just go through people's codes, you grab some things, and you try some things. Why don't we go and stop there? I'm going to continue messing around with this. Again, if you are doing the Honors Embedded course or your grad student, stick around. Stephanie, you and I are meeting. Why don't you come back in maybe like 10 minutes? Unless you want to stick around, totally up to you. Is that good? Okay. Everyone else, ladies and gentlemen, have a lovely afternoon. I will see you all on Thursday. Excited and happy for the exam. Oh,
Professor,
and by the way, the exam went up through last Thursday's material. It does not include today's material.
Professor, I have a question.
Fine.
I work Thursday at EMS and I haven't been able to get the swap. It's okay if is it okay if I take the exam at work, but if I get a call or something?
Yeah, or just tell me when it would be a better time for you to do the closed book. We'll figure out. We can make something work. If you want to do it then, and you don't get a call and you can finish it, great. If you want to do it then, you get a call and you have to come back to it, just give me a heads up. You know,
Okay.
And you can give me a heads up after the fact if you got to get going.
Okay. Okay, sounds good. Thank you so much.
You got it. Any question? Alana. Ipti, Logan, Rotilla, we're talking Honors Embedded?
Yeah.
Okay. Rotilla, you doing Honors Embedded too?
Actually, we haven't talked about it, but I'm just here to discuss it. I'm not sure if I had to, if I needed to start from the beginning of the course or if I can do it now.
Oh no, I don't start it until now.
Okay.
This is exactly when we get going with the Honors Embedded experience. Because what I want people to do, and Mike and Forrest, you guys are including this, is either I want you to bring me a data set that you want to explore. Because what we're going to do is we're going to do an open-ended exploration to answer some questions with a data set. If you have a data set you want to explore, and I think, Mike, you do have a data set you want to explore, then use that.
Oh, yeah.
If you don't, then either we could use something that someone in the group has, or I have a bunch of data sets that are not fully explored that we can pursue. What we'll be doing is basically taking open-ended data and using it to identify some research questions of interest, to modify or manipulate the data to get it to work in R, to do the analyses, to create the figures and write up a short summary. This Honors Embedded experience normally does not get done by the end of the semester, because in some ways you need to know the different tests and procedures to make decisions about what you want to do with the data. So For the honors people, normally you're giving this to me anywhere from a few weeks to a few months afterwards. And that's fine for honors, that's fine for me. If you are graduating this semester, we will just make that work for you so you can get it done in a timely fashion. Boris and Mike, I want you guys to do it as part of the grad experience. Again, same thing. It's not due by the end of the semester. Instead, you'll just give me a time frame from when you're going to be able to give it. And for you two, I think you either want to use data that you have or data that someone else has already collected that's aligned with the type of